Thank you for that introduction, Stuart. I'm Dr. Nicola Kingswell and I'm the Scientific Director here at Labskin Limited, and I'm going to talk to you today about how we at Labskin have developed a new in vitro 3D human skin equivalent model to model atopic dermatitis. So what is atopic dermatitis? It's a chronic relapsing inflammatory skin disease characterised by epidermal barrier dysfunction, immune dysregulation and epidermal spongiosis. Clinically, atopic dermatitis manifests as dry, pruritic, inflamed skin, often accompanied by redness and visible raised and flaky lesions. These symptoms arise from a complex interplay between genetic susceptibility, environmental exposures such as to allergens or chemicals, immune responses and alterations in the skin microbiome. Approximately 15 to 20% of all children and 1 to 2% of all adults globally are affected by atopic dermatitis at some point in their lives. And atopic dermatitis can improve as the patient ages, but for some the disease persists all throughout their lives, having a profound effect on their quality of life, including intense itchiness, disturbed sleep, social stigma, poor self-esteem and psychological distress. The epidermal barrier is essential in skin to prevent water loss and ingress of allergens and bacteria. In atopic dermatitis, the epidermal barrier is compromised. Filaggrin, a structural protein essential for barrier integrity, and in 50% of all atopic dermatitis patients, they have a loss of function mutation in their filaggrin gene. Claudin is another important protein. It's a tight junction protein critical for paracellular permeability. Reduced claudin expression has been noted in atopic dermatitis patients with genetic variation and disease severity. Loss of tight cell junctions results in disrupted skin barrier function, promoting inflammation and allowing microbial invasion. In the acute phase of atopic dermatitis, Th2 responses dominate, resulting in elevation of IL-4, IL-5, IL-13 and IL-31. These cytokines drive B-cell switching and IgE release, eosinophil activation and suppression of epidermal differentiation and barrier protein expression, with induction of pruritus and inflammation. Persistent cytokine signalling and barrier disruption results in chronic atopic dermatitis, characterised by epidermal hyperplasia, altered keratinocyte differentiation and sustained inflammation. The barrier impairment initiates and perpetuates immune dysregulation. The skin microbiome plays an important role in overall skin health and immune balance. In atopic dermatitis, Staphylococcus aureus has been shown to be associated with flare-ups and severity. A compromised skin barrier allows Staph aureus to penetrate deeper into the epidermis, where it outcompetes the commensal microbiome. Staph aureus itself produces toxins and superantigens that further amplifies the immune activation, enhancing the characteristic inflammatory cycle of atopic dermatitis. Despite advancing the understanding of atopic dermatitis, effective therapies remain elusive. So mouse models fail to mimic the human skin structure, barrier function and immune system. Ex vivo skin implants undergo rapid degradation, making them ineffective for long-term studies and microbiome interactions. Therefore, there is a critical need for a living human relevant skin model that can faithfully recapitulate the structural and molecular and microbiological features of atopic dermatitis. Such models would enable mechanistic studies, host microbiome interaction analysis and robust therapeutic testing. So what is Labskin? We are specialists in skin science and microbiology. We have a 3D human skin equivalent full thickness model that fully supports the microbiome. It was originally developed by two microbiologists at the University of Leeds who wanted a really good medium for studying the skin microbiome. They didn't want to just look at microbes in isolation on agar plates or shake flasks. They wanted something they could create these commensal co-cultures. The technology has been developed over the past 16 years with more than 30, probably nearer 40 scientific journal publications now. And we work with industry leading skincare, cosmetic brands to support screening and claims. We also work with wound healing and novel therapeutic developments. Labskin features a living, metabolically active skin equivalent with dermal and epidermal components. It's constructed from primary human skin fibroblasts and keratinocytes. We use a fibrin-based dermal scaffold. So we seed the fibroblasts into this gel layer to create the dermis, and the fibroblasts produce their own extracellular matrix, including de novo collagen. And then we seed the keratinocytes onto the surface, and we create this differentiated, multi-layered stratified epidermis with a full thickness stratum corneum. We can produce the model in quite high numbers, so we get high quality and reproducible numbers for statistical analysis. And it is reactive, so the keratinocytes will produce cytokines when they encounter things they don't like. The top surface is dry, just like human skin, which provides a physical protective barrier, but also allows us to apply things directly to the surface, whether it's microbiome or products. It can also be used for transepidermal penetration and skin localization studies. And importantly, because the model produces its own antimicrobial peptides, we can look at microbiome interactions. And it does behave like skin, so it expresses key markers such as Ki67, filaggrin, claudin, kallikreins, cytokeratin. And this is what it looks like. So if we take a slice through Labskin, you can see here that we have a fully formed dermal layer where the fibroblasts are busy releasing that extracellular matrix and collagen, and then you can see we have a fully differentiated epidermis. So we have a fully formed stratum basal, and the different layers going up to a full thickness stratum corneum. And this is how Labskin compares to human skin. So on the left of the slide there's a human explant, and then in the right slide there is Labskin. And then in the bottom we've done some staining for cytokeratin, filaggrin, laminin and procollagen. So you can see the expression levels and the structure of the skin is very similar. And importantly, with the Labskin model, we do have these antimicrobial peptides. So here we have beta-defensin and RNA7. And as I've mentioned previously, we can populate Labskin with microbiome. So we can colonize it with bacteria, with fungi, with viruses, both in monoculture or co-culture. We can look at defensive properties. We can look at microbial challenge. We've created our own mixtures. We call one a 3X mix, which is a mixture of C. striatum, C. acnes and S. epidermidis. These are the top 3 microbes that make up everybody's microbiome, just in different ratios depending on who you are. And we also have a scalp mix, which is this 3 mix of skin microbes plus Melissetia. So this is the yeast or fungi that can cause dandruff. And here's just some nice immunofluorescence imaging looking at the microbe on Labskin. So here we have the microbial layer on the outer surface of the skin, and then we've stained the skin for expression of filaggrin and desmoglein, and the cell nuclei are also stained. And if we dig a bit deeper into that imaging, here you can see the bacterial layer in blue on the surface of the skin, and then you can see that filaggrin expression between the cells of the epidermis. And this is a study that we did with the University of York, where we added Staph aureus onto the surface of Labskin and did a bit of a time study to see how quickly an overload of Staph aureus would penetrate into the skin. And interestingly, here on the middle panel, you can see the Staph aureus here in purple that's penetrated into the skin. So not just penetrating between the cells in the layer, but actually into the skin cells themselves. So we wanted to create a mimic of atopic dermatitis using our Labskin model. So we've nicknamed it Labskin-AD for obvious reasons, but we wanted to ensure that we could model the phenotype, the immunological reactions and the microbiome of the Labskin-AD, but also look at therapeutic interventions. So we know that in atopic dermatitis, the skin has a disrupted skin barrier. So we wanted to know if we could create this trademark Hawley appearance in the epidermis associated with the loss of tight cell junctions and barrier function. We initially tried 2 methods, applying a known chemical irritant into the media and also using a cocktail of cytokines that are known to be elevated in atopic dermatitis. This slide shows that both methods can induce the epidermal spongiosis, but that the cytokine cocktail appears more controlled. We looked at the expression of key proteins in the skin barrier, so we looked at desmoglein, filaggrin and claudin, and also psoriasin, and compared the Labskin disruptor skin barrier models to ex vivo human skin and normal and healthy Labskin-S. Both the disruptor barrier models showed the loss of tight cell junctions, but importantly expression of psoriasin. So we knew it could be possible to create a Labskin atopic dermatitis model. So we focused solely on using the cytokine cocktail. Labskin was produced as per our standard SOP for producing normal healthy Labskin. At 12 days air liquid interface, a cytokine cocktail was introduced into the culture medium, and this was incubated for a further 72 hours, and this showed disruption of the epidermal barrier. So the top row here, this is the Labskin-AD, so this has had the cytokine cocktail induced, and the bottom row, this is just normal healthy Labskin. So you can see here, after 72 hours, we are seeing that epidermal spongiosis. So the takeaway from this slide is that the cell-cell junctions are disrupted in the Labskin-AD model. So then we refined the protocol further, and then we wanted to look and see if it was possible to visualise the key skin barrier proteins in the Labskin-AD model and how they would compare again to normal healthy human skin and our healthy Labskin-S. From this slide you can see that we looked at expression of desmoglein, filaggrin, claudin and psoriasin again. It looks to be a down-regulation in the expression of claudin, but a very marked upregulation in the expression of psoriasin. And then we wanted to look at the interactions with key skin microbes. In particular, in AD patients, the microbiome profiles show an increase in S. epidermidis and S. aureus. So we used a 3 mix of C. acnes, C. striatum and S. epidermidis and enumerated colony forming units after 24 hours. And we did a monoculture with the Staph aureus, and again we enumerated after 24 hours. And from this graph you can see that both S. epidermidis and Staph aureus showed an increase in colony forming units after 24 hours in the Labskin-AD model, and this was statistically significant compared to normal healthy Labskin. So we know that we can create a Labskin-AD model, a nice in vitro skin mimic for atopic dermatitis. But could we reverse that epidermal spongiosis? So we treated Labskin with the cytokine cocktail, and then we introduced a JAK inhibitor, upadacitinib, was applied. And this showed that this JAK inhibitor was able to reverse the epidermal spongiosis after 96 hours. So if we just focus in on these 2 slides here, this is the atopic dermatitis model without the JAK inhibitor, and this is the atopic dermatitis model when it's been treated with the JAK inhibitor. So you can see we've almost got this reversal of the loss of cell gaps. So we wanted to dig a bit deeper into these protein changes and pathways and the alterations. And we've previously been working with the proteomics team at Leicester University, looking at our standard Labskin models with and without treatments. And using their very fancy mass spectrometry systems, we were able to show over 6,000 proteins that are expressed in the standard Labskin model. So we were interested to compare the Labskin-AD model to the Labskin standard model and see which pathways were activated, which proteins were changed, which cytokines were activated. So I'll now hand over to Colleen to take you through the work that's been performed at Leicester.
Hi everybody, I'm Colleen Maxwell, a postdoctoral research associate in the Van Gies Multi-Omics facility at the University of Leicester. Today I'm going to show how we're using next generation proteomics to gain insights into Labskin models. To begin with, just a little bit about us at the Van Gies Multi-Omics facility. We're based at the University of Leicester, and most of our research is on the translational biomarker front. We bring together cutting-edge mass spectrometry and multi-omics technologies to turn complex biological data into insights that matter for patients. That includes proteomics, metabolomics, and lipidomics. Our aim is to uncover molecular signatures which can improve diagnosis and monitoring in major diseases, including cardiovascular, cancer, and respiratory. I just want to mention as we begin as well that no collaboration happens alone. My PhD student Nikita and my postdoc colleague Ellen have been working on this project with Labskin as well. So today I'm going to focus on proteomics, which is the large-scale study of proteins in biological systems. Proteins are the functional molecules that drive biology and disease, so by measuring them at scale, we can uncover disease mechanisms, get a more detailed picture of underlying phenotypes, identify biomarkers for diagnosis and prognosis, discover therapeutic targets, and ultimately enable precision medicine. In biological research, the goal is to move from molecular discovery to patient benefit or to benefit consumers, and to drive that, we want to measure as many proteins as we possibly can. The technology we use to measure global proteomics in really high throughput for this purpose is called mass spectrometry. And that's where my expertise comes in. With mass spectrometry, we can measure thousands of proteins at once, and my role is to ensure that the technologies we use are cutting edge, scalable, and robust. And that means developing highly automated workflows, improving analytical methods, and innovating in data analysis, all of which we've used in this collaboration with Labskin. And the potential of proteomics in the Labskin model is massive. In this particular case, we wanted to further elucidate or validate molecular changes underpinning the observed Labskin-S and Labskin-AD phenotypes. But of course, we can also use proteomics to assess the impact of treatments on the global proteome. How do we do it? So I'm going to walk you through our workflow here a bit. So the first step is to homogenize the Labskin samples, and we do this to disrupt cell membranes and tissue structures to release the proteins. And we do this by beading the Labskin in a tube with zirconium beads in ripalysis buffer. Next, we measure the protein content of each sample with a colorimetric assay, and we normalize the protein content across all samples to the same concentration to avoid introducing bias. We do this with our liquid handling system, Andrew Plus, which is a very flexible liquid handling system that's essentially a robotic arm. You can see it operating here in the video, and it's great because not only does it save you time, but it also increases accuracy and longitudinal reproducibility compared to doing the protocol manually. Next, we swap to our other robot called the Bravo, and we perform a protocol called SP3. And this protocol removes residual contamination from small molecules that we're not interested in and the RIPA buffer. And to do that, it uses magnetic beads. And then it digests the proteins on the beads into peptides using the protease enzyme trypsin. The reason why we want to look at peptides is because measuring big intact proteins is actually very analytically challenging. So instead, we break them down into peptides and infer the presence and abundance of proteins in the sample based on their constituent peptides. And here's the Bravo robot in action. It's a very high throughput, very precise robot, which can prepare hundreds of samples per week. And it's really good for multi-omics as well, so you can actually use it for metabolomics and lipidomics in addition to proteomics. Next, we load evotips, which are just the storage vessels for the peptides before they're introduced into the mass spectrometer. Then we analyze the sample using liquid chromatography coupled to mass spectrometry. And then we use downstream bioinformatics processes to calculate the protein abundances and determine which proteins are different between the different types of samples. So just to tell you now a little bit more about our mass spectrometer. So just last year, we were able to get a new next generation liquid chromatography mass spectrometry instrument called the EvoSep, coupled to the timsTOF HT. And compared to our older mass spectrometer, which was still really good, but this has revolutionized proteomics for us. We have very low acquisition times of 10 to 20 minutes. We can use tiny sample amounts to start out with, and our depth of proteome coverage has improved over 5-fold. So here it is in our lab. I know it doesn't look very exciting, but this is an excellent analytical instrument, and we've been using it for analysis of all the Labskin samples. So we analysed Labskin-S and Labskin-AD samples following the workflow I've just described, and we identified 6,613 proteins using just a 20-minute acquisition time. And then the questions we wanted to ask are, which proteins are differentially abundant between the two models, and what do they tell us about the phenotype? So the first analysis we did was unbiased clustering techniques to determine if the global proteome profile can differentiate between Labskin-S and Labskin-AD. So as you can see from this principal component plot, there's a big separation here, both on the first and the second principal component between the two models, and that's just based on their protein profiles. And this is also seen when we perform hierarchical clustering analysis, which reveals the Labskin-S samples clustered together here in green, as do the Labskin-AD samples. And you can see if you look at the heat map of protein expression, that the protein expression is different between the AD and the S samples based on the global expression profile of the proteome. The next question we ask are, which proteins are driving these observed differences in phenotype? So we performed a differential expression analysis to determine which proteins are statistically different between Labskin-S and Labskin-AD. And we uncovered a total of 1,277 proteins which are differentially regulated. And you can see here in the volcano plot, it's these proteins in red here which are statistically different, and the ones up at the top corners are the ones which are the most different between the two models. Those on the left are the 707 downregulated proteins in the Labskin-AD model, and those on the right are the 570 which are upregulated. So if we just focus on the proteins which are upregulated, so those which are more highly expressed in the Labskin-AD model, we can see, as we would expect, that cytokines such as interleukin-19 are upregulated in the AD model compared to healthy, and also members of the JAK-STAT pathway such as Janus kinase 1. And interestingly, Claudin-1 expression was significantly elevated as well. So Claudin-1 is linked to AD susceptibility, but a cytokine-induced upregulation may represent a compensatory response aimed at restoring barrier integrity. And this highlights the dynamic and context-dependent regulation of barrier proteins in inflammatory skin disease, and that we can capture this dynamic response using global proteome profiling. And if we take all of the upregulated proteins together, we can see which pathways they're members of, which are upregulated in the AD model. So to highlight just a few relevant pathways, we see lots of interleukin signaling, we see lots of indicators of chronic inflammation and immune response, extracellular matrix and collagen degradation, and as mentioned, JAK-STAT signaling as well. And now let's look at the downregulated proteins. So these proteins are lower in the AD model compared to Labskin-S. There were 707 in total, but to highlight a few, we have filaggrin 2, involucrin, and keratin-1. And these are all reduced in the AD model, and this implies a reduction in late-stage keratinocyte differentiation. And this is confirmed by pathway analysis. We can see substantial disruption of keratinocyte differentiation and RNA processing pathways, which are consistent with impaired epidermal maturation and reduced transcriptional activity. So that's just a little taster today of how we can use unbiased mass spectrometry-based proteomics to gain really deep insight into the molecular phenotype of the Labskin model. We're currently doing ongoing work looking at the impact of treatments on the model and whether certain treatments can soothe that dysregulated proteome that we see in the AD model and move it back towards that healthy state or stop it from progressing in the first place. And with that, I would like to thank Labskin, especially Stuart and Nicola, and my colleagues at the Omics facility, especially the group director, Professor Don Jones, my PhD student Nikita, and my colleague Ellen. Thank you for listening. I'm happy to take any questions.